All right, so welcome everybody to another episode of the Future Leaders in Mechanobiology seminar series. Today's speaker is Kateryna Fal. She's currently a senior postdoc at the Chromatin Dynamics and Developmental Transitions team in Grenoble, and it's CEA Grenoble, which I understand is sort of like our DOE. Is that right? It's like a government institute.
Yes, it's a Centre de Géotonique. So
yeah, I was scared to try to pronounce that. Anyway, Kateryna is actually an old friend of our lab. We collaborated with her in order to understand the role of the PAF1 complex in mediating chromatin remodeling way back when she was starting her postdoc in Olivier Hamont's lab. She was, of course, extraordinarily productive during that work and has continued to really expand on what we know about chromatin remodeling complexes and their role in mediating mechanical signals, and is now expanding way beyond that to understand chromatin remodeling in response to mechanics in general. And so that's what she's going to talk about today. And I'm just going to hand it over to her now. So welcome, Kateryna.
Thank you so much. Thank you for the invitation. I'm so happy to be here today, even virtually. I'm going to share my screen. And I will, yes, I hope it works. So yes, I'm a postdoc now in Grenoble, a beautiful city surrounded by mountains. It's been a while since I started the postdoc, and it's been a while since I've been changing topics, but through all this, the general red line was chromatin and what does it do for plant development. It doesn't work now, it should work. So plants, they need to react as any living being to the impacts of the surrounding. They have to adapt. If there is not enough sun, they are growing. If there is more than enough sun, they have to survive. As we see for the reactions of the plants which are exposed to wind, they are sometimes shorter, and those who are not, they are a bit longer. So there are developmental mechanisms which are involved in those regulations. So plants which can be genetically identical, when they're exposed to different environmental conditions, they become different. So this is what is interesting. So what determines those differences? And in all the beings, when they are identical genomes, when they're being exposed to environmental factors, what happens is there are some differences appearing in the phenotypes, and those differences are due to the epigenetic differences. Even in the human beings, when we compare the epigenomes of those from 3 years old and 50 years old, due to their life factors, due to their life conditions, we see that there are differences occurring. As in mice, here there are 2 mice, a classic example of DNA methylation in the agouti locus, and the fur color is different. And in plants, we see that as well, from the time of flowering due to exposure to cold to their holly leaves shape due to exposure to touch, to the mechanical stress. And here we come to basically the point that the reactions, they occur due to changes in the gene expression, which lead to changes in the metabolic reactions, but those happen after nucleus perceives that signal. So nucleus and ultimately chromatin, they react to the signal which comes to the cell, and they mediate that reaction on a metabolic level. And chromatin, what is actually chromatin? That is the DNA plus the proteins around which it's wrapped. And also it comprises the modifications, the epigenetic modifications, which I mentioned just before. So those which I will be mentioning most, they concern the modifications on the tail, so on the sides of the proteins, of the histones, around which the DNA is wrapped. And those are quite mobile. Some of them, when they occur, they permit the chromatin to be a bit more relaxed, and those are permissive marks. So it's more accessible for the transcription factors, and though the genes which are located in those regions, they are more actively transcribed. And then the others are more repressive, so they permit the condensation of the chromatin, and then the genes which are located or happen to be located in those regions, they are less accessible for the transcription factors, and they are so-called repressed or less transcribed. And these transitions, they are occurring quite in a mobile way. They are flexible. The chromatin is breathing, and that is happening also in response to the changes in environmental conditions. And what there's a famous circle, and Olivier Amand, with whom I was working before, is always shows this. There's a feedback loop from the mechanical signals to the cell shape and to what is occurring and feeding back. But then if we expand it, what happens is mechanical forces, they of course, they impact the cell growth, not only plants and animals, and the differentiation has also been shown. And then of course, it has consequences for gene expression, and gene expression is linked with the chromatin status, and chromatin status is linked with the epigenetic marks. But then what is less known, especially in plants, is how these mechanical forces, do they have a direct connection, a direct signal towards the chromatin organization or towards their changes in epigenetic marks without passing through the cell shape and so on and so on. So can the application or changes in the application of mechanical forces directly change the epigenetic marks or directly change the chromatin status, which leads to changes or reorganization in the epigenetic marks and thus leads to the changes in gene expression? So where is this link? Where is this signal? And my story starts with Arabidopsis, with a project which was initiated by Elizabeth Haspel, here present. And she performed a screen where they were trying to identify plants not reacting to touch. As I mentioned before, plants which are touched, especially Arabidopsis, if they're touched before flowering, then they don't grow as high. And then by screening many dozens, hundreds of lines, they managed to find a single one which was not reacting to brushing, which was growing as high. And amazingly, that was the mutant of the chromatin factor, which is BIP3, which is a subunit of a complex which guides the RNA polymerase. So here comes chromatin. It's not directly linked to the cell wall or to perception of the signal, but rather to what happens inside the nucleus after the signal goes through. And what is PAF complex? So it's the complex which is really conserved in yeasts and in animals and in plants. In plants, it's, as often happens, subunits have different names because they were all discovered in a screen for early flowering. Here, absolutely not related to our story. Then they have, what they share is the common features of the phenotypes. They're all small, and the strong alleles of these mutants are really affected in development. However, here, the weak allele, the only phenotype which was detected is this touch perception defects. So PAF complex, it mediates the initiation of transcription, but it also mediates the RNA degradation after RNA is not needed anymore, the transcripts. And it's also involved in targeting of the histomethylation, and it's connected with the activity of the methyltransferases for, especially for H3K4 and H3K36 methylation, which are active marks on the H3 histone. So which are marks for active transcription of the genes which are located nearby. So what on my side, what we did is to test the expression of the genes which are known to be upregulated after the touch in Arabidopsis, after we touch the plants. So we sown some seedlings and we touched them with a brush, and we took the samples 30 minutes later, and then we did it again and we took the samples 30 minutes later. I tried several times, meaning I continued after 3 or 4 touches, but then the seedlings started to be a bit fragile and wounded, so we stopped after 2. And what is interesting is after 2 touches, the genes, like touch 4 gene, it still goes up. So it is strongly induced in the wild type after single touch, but it still can be induced in the mutants. It just needs a delay, it needs to be pushed stronger. So the mutants are not deficient in a reaction, but they require stronger stimulation. Of the active histone mark around this touch 4 locus, which we detected. This is only one of the features of this mutant. So it's delayed to the reaction of the surrounding. It was also observed in a larger phenotype, especially in a stronger allele phenotype, where the geometry of the plant itself is a bit more affected. And for example, in nature, it occurs that the seedlings have more than 1 cotyledons, more than 2. Sometimes they have 1 more, 3, but in nature, it's really rare. Well, 3 really often has that. It's up to 20% of plants in the population. What is funny, it depends on the environmental conditions. So when we place the plants in different growth environment, we can stimulate. So it's inducible phenotype. We can push plants a bit more, and then we obtain more. And also, the organization of the calyx along the inflorescence stems, it's really strict. And usually in Arabidopsis, between each consequent calyx which appears on the stem to each consequent flower, they grow in a spiral going up. There is 137 degrees. It's a canonical angle. And this organization is called phyllotaxis, the organization of the organs around the inflorescence stem. It's really normalized. So this peak, it's a frequency of the distribution. So if we measure all the angles along the stems of several plants, I went to up to 50 plants, and we plot them all, of course, this spiral, see the spiral going one direction, it can change and it can go different direction. And then we have this little angle, little second peak. However, generally, the most of the angles, they fall in this category of 137. Well, in the mutant, when it grows in the long day conditions, it's pretty much the same. However, if we place it in a lower temperature, we allow a plant to become a bit bigger, to accumulate a bit more biomass. The meristem becomes a little bit bigger, and then it makes more mistakes. And then we see this distribution of the angles getting wider. And with the help of Yassine Rafai, who is now in Rennes University, he was working with Henrik Johnson in Cambridge at the time, did some modeling to predict how many perturbations of the sequence of the calyx should occur for this to happen. And basically, it is a noise. So it's not just aberration, it's much more complex than that. It goes into, it's a chaos. So this only occurs in certain conditions. In our case, it was 16 degrees and the continuous light. But then when we started looking at those plants in more details, the same conditions stimulated the flowers to, to be less sterile, because usually they are nearly sterile, but at the same time to produce flowers inside the flowers. So the flower meristem, which should be terminating at the point to make the carpel, to make the ovules inside, it's not terminating. And inside the flowers of the mutant, we can sometimes observe the whole stems with flowers developing. And if we did some sections of the calyx, it's like a Russian doll. It's the one inside the other, inside the other. Sometimes we see the whole flowers, branches with the flowers developing inside, which is pretty amazing. And also interesting that by placing plants from 21 degrees short day to continuous light, we obtain that. I'm not showing the just long day, because in long day condition, it doesn't happen. It has to pass through the short day and then to either continuous light or the long day to produce this effect. We suppose that it's because the meristem grows a bit bigger and then the plants slow down in development, and this slowing down permits to do more mistakes because the meristem is larger, so the field of mistakes possibilities are larger. But then when it goes to continuous light and the same temperature, it doesn't go, basically at the end of the meristem, at the beginning it does mistakes, and at the end it still does mistakes about the same rate, which is also interesting because in Arabidopsis, this flower indeterminacy phenotype or other like fertility problems, they are sometimes occurring at the beginning of the production of the seedlings. The first flowers, we don't use them for the crosses because they are not as fertile, as good as the next ones. But then it goes down and in the middle it should be fine. However, here it's a second wave appearing. And so basically we tested some agamous expression, which is a marker for flower meristem termination. And in the mutant, in some of the flowers, it's lost. I just say some because it's not 100% penetracy. And our hypothesis is that PAF1 complex, the same as with the phlotaxis, it acts in filtering this transcriptional noise and protects from making the mistakes. So when we take off the PAF1 activity, or perturb the PAF1 activity, plus we challenge the plants with specific growth conditions, we provoke these mistakes, we push it to the threshold where it has to make this trade-off. Plants have to become smaller to protect themselves from these events occurring. And then if we place them in a lower temperature plus longer day, here we go. So this actually is an evidence of this protective role of the chromatin in the developmental programs and of the connection between the perception of the environmental conditions for plant development. And so what is here, I'm jumping a little bit. So what is special about plant cells? We know that mechanical surrounding of the animal cells that can impact the cell specialization, the cell lineage determination. So what happens for the plant cells? They have the walls. We know that the plant cell wall is really involved in the perception of the mechanical signal, in the transduction of the signal. And the shape of the plant and shape of the nucleus inside the plants, inside those cells, it depends on the shape of the cell and the shape of the everything inside, and the nucleus is not disconnected from it. And moreover, because of the cell walls, plants cannot escape. They have to cope with the occurring stress. So in plants, there is an interesting established system for observing the developmental processes. Is the pseudapical meristem. And here, for the next part of my talk, I will describe how we try to use it as a model for addressing the questions of how nucleus perceives the signals of the mechanical stress in this context, in the tissue context. So pseudapical meristem, right? It's in the middle, it's on the top, there are cells here developing, but then there are organs appearing around, and organs appear in a spiral manner, which basically, as we suppose that they are nearly identical or as identical as they can be as their flowers, they have this repetitive pattern. So we can follow their same stages of development, and we can make a time course, and we can image those meristems in a confocal microscopy. We can follow their developmental and development of the organs and development of the boundary domains. Why the boundary domain is so interesting is because there's a directional stress occurring when the organ separates from the meristem itself, and it starts to grow as an independent entity just next to it. And it has been shown that the microtubules in the cells which are trapped here in the middle, they align along the vector of this stress. So the cells are stretched, but at the same time they are compressed. I will talk a bit later about it. So this anisotropic stress here, there's this question of the cells. So what happens in those cells? What is interesting is that not only those cells are deforming, we know that there is a pattern of expression of genes which are associated specifically with this domain. And there are genes which are expressed only in the central zone, right? There are those which are expressed in the organ and in the meristem, but not in the boundary. But there are also those which are expressed specifically in the boundary, like the STM, like the CAC group of genes. There are several others. And for example, the CAC genes, the expression occurs in parallel with how the boundary forms, and the pattern slightly changes with the boundary getting deeper. So here is just a snapshot at 0 and 24 hours later of the same organ, where the boundary here is when it gets more deeper shade of blue, it means that the boundary getting deeper. So the organ grows and then it folds. And here is the expression of CAC3, which is associated with this domain. So first it's more in the middle of the organ boundary, and then it goes a bit more to the sides and getting deeper. So why there? And is it because they are transcription factors just expressed in the same domain and they are regulating those genes, but then chicken and egg again? So why transcription factors are there? So what is special about those genes and why there? And what is special about those cells? So to address this question, it was during my postdoc with Olivier, we tried to follow the deformation of the nuclei at the organ boundary. And I followed organs of several pseudoapical meristems from time point 0 to 36. It's several time points, every 12 hours. And so the boundary is getting deeper. So organ is getting bigger. And if we look at the nuclei here, they are getting more deformed. We had incredible luck to get our hands on the generated reporter line with the SUN1. It's a protein which is anchored to nuclear envelope. It's used to YFP. So here we are seeing the contours of the nuclear envelope. And it's incredibly stable. So we could actually follow the same nuclei through several time points, and they are live. So next point, we decided we collaborated with Henry Johnson's group and with Jose Teles and Nicolas Korsbo. We developed a pipeline to analyze the nuclear shape and to correlate it with the form and with the development of the boundary. And so what we found, assessing the aspect ratio. So when I will be talking about aspect ratio here is in 2D. So we project the nucleus and then we assess the ratio between the longest and the shortest axis of this ellipse. And the curvature, basically, it's a projection of the curvature of the organ and the boundary. And when it's in blue, it's negative, it means it's a saddle shape. And when it's in red, it's something which is bumped, it's a positive curvature. And so what we found is that there is a small subpopulation of the nuclei, which are right in the middle of that blue deep boundary, which are really highly deformed, which are really highly elongated. The high aspect ratio, which is over 1.5. So that's a threshold for us. And but there are only a few. So in every single event, not all the nuclei which are in the boundary are getting deformed, but only a few, which gets it a bit harder to analyze. We need to pick. But then again, why are they deformed? Are they squeezed or are they stretched? And why is it important to know that? Well, it's because if they are connected to the cytoskeleton, they are connected to the nucleus shape. So if cytoskeleton is really important for the maintenance of the nuclear shape, which is logical because nucleus is connected to the cytoskeleton through the nuclear envelope, through all those complexes which are anchored to it, and they are anchored to the chromatin from the other side. What is important to know about plants is that unlike in animals, this a little bit more difficult. There are no lemming proteins per se. They are proteins which are likely to have the same function, and their complexes anchor to their nuclear analog, which have the same function. However, they're not identical, they're not homologous to those in animals. And the link complex, the sun is one of those, is exactly, it's a link into the exterior interior of the nucleus. And what we used is the mutant of their crowns, crowded nuclei, which is incredibly interesting because the plants themselves on a macro developmental phenotype, they don't display anything. They develop normally. However, when we look at the shapes of the nuclei within the young leaves, well, they are different from the wild types, definitely. However, the cell shapes, they are really slightly perturbed, they are still elongating. So why, when we disconnect the nucleus from the cells, the cell continue growing, the plant continue growing, and the nucleus stays disconnected. So the signal still goes through. So it means that the nucleus shape is not important. But then when we look, this is in leaves, when we look at the meristem, the situation is different, because in the meristem, the ratio between the volume of the nucleus and the volume of the cell itself is not the same as in leaves. The nucleus takes almost the whole volume of the cell. The vacuole is really pushed on the side. So when we take off that connection, well, the effect is not so drastic. So when they are nearly the same. So when I try to put the numbers on those changes, the nuclear area is maybe, this is in the meristems, they're maybe a little bit different in a crown mutant comparison to the wild type. However, of course, the circularity is changed. They are more closer. What circularity is, the closer we are to the circle, the closer we are to one. The more different we are from the circle, the more different. And then the aspect ratio, right? So aspect ratio here, they are nuclei, all the nuclei around the boundary plotted. So some of them, of course, as I mentioned, only the small fraction of them will be more elongated. However, indeed, in a crowded nuclei, in a crown, we don't see that many of that fraction which can be elongated. So maybe it could signify that, well, they are not connected, well, when they are not connected to the microtubules, which are elongating the cells, maybe they cannot be elongated. So maybe indeed they are stretched. But when we looked at the boundaries, and we look at the zoom and we look manually, we see that the nuclei, which are, this is a younger boundary, this is a deeper boundary. It's the same organ within 36 hours. So those are nuclei which are getting squeezed. And here I'm doing squeeze because if they are not connected to the cytoskeleton, which is pulling them aside, it means that it's most likely the folding of the tissue which brings them to being squashed. And so, right, we saw that the aspect ratio and the quantity of the nuclei with the aspect ratio in the boundaries, it's lower in the crown mutant comparison to the wild type. However, it's still possible. So both factors, this squeezing and the stretching, are important. However, folding of the tissue prevails over the stretching of the nucleus. And so the physical compression of them brings that effect. So then what is the effect for the chromatin? When the nuclei are deformed, we see that some things should happen. And when we looked, so this is a marker for the heterochromatin, basically. This line was generated by Mathieu Ingouf from Fred Berger lab. And so what we imaged the nuclei at the boundary of the organ and closer to the meristem. And we saw that so what is CG regions are the chromocenters. So they are the shiny regions here, the regions where the chromatin is more condensed. And so they are more regions which are having DNA methylation, so they are suppressed, they are not actively transcribed, most likely, they are not accessible for transcription. And that's logical. The less regions they are, the more chromatin is compressed, and this we see more in the nuclei which are stretched, which have an aspect ratio over 1.5. So here, that's logical, but that's nice. But then what happens to that chromatin? Because chromatin is, as I said, there are many histones, there's a DNA, there are modifications. And so when the chromatin is changing the conformation, there are linker histones like histone H1. They should be changing their disposition as well. And in plants, there are 3 isoforms of the H1 histones, in plants in Arabidopsis. H11 and H12 as those which are expressed independently of the what is happening in the surrounding, and H13, which is has been reported to be associated with the stress. Now, stress in a large sense, because there were reports that it's induced by the darkness, by salt, by drought. Here, I looked at the expression of those histones with the reporter lines in the meristem, and well, it's interesting because H11 and H12 are present in the organs, a little bit in the meristem. We didn't see the strong expression. They should be there. But H13 is in the boundary, and mainly in the deep boundaries. And that nicely fits what we saw with the chromatin condensation, that also fits with the theory that this is a region under stress. And so that's a stress histone isoform. So, but then, so this is the H33, which is, it should be everywhere, and we see it everywhere strongly. So then, after I left already, Juan helped us, and he did the amazing job compressing the meristems, and he managed to induce the H13 expression in the meristems which are treated with NPA, so they don't have organs, because when we dissect the organs, it's wounding, and wounding also induces H13, so we were stuck there. And so he managed to compress the meristems, and this was one by one. It was a really long job. And he managed to induce the expression. So we can, it actually confirms that when we squeeze that tissue with the growth of the organ, we indeed subject those cells to the compression, and it can induce the changes in the chromatin. So of course, then the question, if we remove that histone, what happens then? So in the mutant of H13, the plants do not display strong phenotypes unless they are under drought or salt stress conditions. So what we did, we introgressed a line for the CHH DNA methylation in that in those plants. Why that one? Because previously it has been reported that the regions which are bound by H13, they are the regions which are related to the stress genes, and which are also subjected to the DNA methylation in that context. So what we expect is that the plants which are not having the proper function of H13, which is not having it, they cannot induce the DNA methylation of those regions in the same way, in the same manner. And that's basically what we saw. So in the same nuclei which are elongated, in the boundaries, when in the H13 mutant, we don't see the induction of the signal which corresponds to that reporter line. So, well, it's a confirmation of our suggestion that the H13 is indeed important for the nuclei which are under stress in the nuclear boundary. Then it doesn't answer the question. The biggest question is the biological function, because those boundaries are still there and the organs are still there. And even if we remove the H13 and we remove that function, well, the organs still develop and the boundaries still develop. So the biggest question then is to understand how, what is critical, where is this weak point? So for me, for further perspectives of my further career development and further looking in a scientific sense is to understand where is this link. So what should we touch to better understand the connection between the chromatin and the regulation of the perception of the signal from the surrounding, and where is the biological function of that? And for example, looking at the direct targets of H13, and why SDG8 is a chromatin factor chromatin remodeler, which is involved in the establishment of the active histone modification, the methylation of 36 lysine. Why this one is also because at the same time as we were publishing the PUF1 and the touch perception, the paper showing that SDG8 is involved in touch perception was published. And SDG8 and PUF complex are in contact, they are interacting in this function, in the regulation of H3K36 methylation. And the genes which are under this regulation are luckily involved also in the regulation of chromatin organization for mechanical reaction to mechanical stress. And then, of course, there are data sets, it's just an example, but there are data sets showing several genes which are upregulated by several kinds of mechanical stress, like the touch, but also there are some data sets for sound stimulation and for some vibrations. And I think maybe in the intersection of all those, plus maybe some screening of additional chromatin factors could answer to the question of what are those maybe specific gene regions which are more responsive to this kind of stress, and maybe manipulating further the expression of those regions could give us an answer to their feedback loop or to how this signal regulation happens. And of course, looking at the chromatin marks which are associated with those regions could maybe point out on the direct function of the chromatin in this regulation. So there are several open questions, well, I hope to persevere and to continue working in this domain further. And I would like to thank all the people I've been working with for the last 10 years, including the great, amazing team of RDP in Lyon and Olivier Amand. Most of the people from that picture already moved on elsewhere. Of course, Elizabeth Haswell and Marie-Edith Chaboutet in Strasbourg, as well as a team of Henry Johnson in Cambridge, and especially Juan Alonso-Serra, who took over the project when I left and allowed us to finally publish the story with Olivier. And of course, I also should thank the current group I am in Grenoble, the chromatin dynamics and development transitions team of Christelle Carl, who is taking completely different axes and routes, but story still leads me towards mechanoperception and still on. And the family, of course, the family, especially during the last 2 years.
Great. Thank you, Kateryna. Is everybody so thank you? So thank you for that great talk. Well timed. Does anybody have any questions for Kateryna? I have a few, but I'll hold them on the back burner. See if maybe we have some trainees with questions first.
I'm training in the area of plant biology. I've learned everything I know from Liz. looks like I don't count. Never mind. I'll wait for trainees to go.
Did you said somebody, it looks like somebody has one. I don't see anybody raising their hand. So if you want to go ahead, you are welcome to ask a question, Guy.
Your back burner is much more interesting than my front burner, but I'll go until, feel free to reject my question and go to Liz's later, Kateryna. But first, Kateryna, thank you for being here. I've enjoyed following your work, and it's great to see it presented all in one piece. Beautiful presentation. I've learned a lot.
Thank you.
One thing I'd like to learn as well is, what do you think, or are there roles of the structures on the inside of the cell in manipulating the nucleus? Do microtubules, F-actin, can they provide enough force to actually make an impact on chromatin condensation, or do you need something as big as salt stress, as something osmotic to really impact the nucleus?
Well, it's a little bit what I mentioned in the example of the crown mutant. So when we mutate the connections here, there is no connection or real little connection between the nucleus and the microtubules, so the cytoskeleton and the cell shape. So when we disconnect that, we know, and there were some DAPI staining performed, that the chromatin is a bit disrupted. However, it doesn't have major consequences for the global plant development. So I think, of course, this transmission of the signal is really important. However, maybe not in a direct way of the cytoskeleton communication. Maybe it's more on the signaling molecules which pass through, and more than, I don't know if I'm answering your question.
Oh, yeah, absolutely. Can I ask a follow-on, or if I already? Sure. So a follow-up question is, there are all these really amazing molecular motors inside plant cells in general. My favorite myosin in the whole world is inside plants. It's really fast. Are they powerful enough to be able to perhaps is it possible for internally generated forces from those proteins to possibly impact the nucleus in a way that would affect outcome, or is it really the outside world that matters?
Well, I'm not the specialist in microtubules. I'm more on the inside of the nucleus. I would say, well, plant cells are not moving. It would be fun to make them move, but maybe because of that, and because of the presence of the vacuole, which takes a huge space in the cell, and it basically pushes everything on the side. I think the osmotic balance has a huge impact on what is happening inside the cell, and then it's probably hard to determine, to take that out of the equation and to determine, okay, that's motors, and that is.
Super. Thank you very much.
All right, Ram, you have a question?
Yes, I do. Thank you. I hope you guys can hear me. I was having some computer trouble, so I'm using my phone. Okay, good. Yeah, thanks for that lovely talk. I was actually thinking along similar lines to what Guy just asked. So I'll sort of ask, I guess, in some ways, a follow-up question, because you mentioned, and I was thinking about these data that you're just showing here on your screen right now with the CROWN4 mutant, where the nucleus is, for the most part, I guess, uncoupled from the cytoskeleton. So I guess I had a two-part question. Did you directly assess, what did you do to sort of experimentally determine how uncoupled the nucleus was from any cytoskeleton connections? And second, I'm really intrigued by the idea of, where you have these fairly significant nuclear shape differences between wild type and the CROWN4 mutant, and yet the plant seems to be okay, at least under your regular growth conditions. Did you check if nuclear transcription factors accumulate differently or something like that, or the kinetics of their accumulation, sort of what you just sort of alluded to in response to Guy's question?
Right. So that would be really interesting to do, especially on the point of, from my point of view as a postdoc with a short amount of time. These are sort of questions which, yes, I wish. Unfortunately, we didn't access that. And there's amazing work by Chieko Goto, who worked on the combination of the Kaku and CROWN mutants, and she did a lot of stuff on the characterization of the nucleus shape in those plants. What I can say is that the CROWN4 is on the CROWN, there are several of them, and to achieve the major developmental effects, we need to mutate several of them. So they are partially redundant. And CROWN4 is interesting in the sense that it has effects on their configuration of their chromatin inside the cell. However, no major effects on the outside of the phenotype. So we didn't go further because for us it was the model for nucleus shape. We wanted to just basically check if it could be compressed in the boundaries still when we are partially uncoupling. So in my opinion, it is partially uncoupled. It's not completely, of course. And maybe that's why the triple mutants are so nearly not viable, because when we completely sever this connection, then of course it doesn't go. So we have to stay partially connected, but then not that much.
Ram, did that answer your question?
Yes, I guess could you speak to whether anyone has tried looking at transcription factors, or even, I'm just even curious what your thoughts would be, like what would be the kinds of transcription factors that would be worth maybe considering if one were to want to do this experiment?
Well, I think that would be really interesting to to assess. For example, I would love to look. Well, my set of chromatin stuff. So I would like to look at the chromatin, basically distribution first of the chromatin marks, and of the distribution of the histone marks, and to see if, for example, already the organization of the chromatin can be different, and then to see the chromatin regulators, like those which are involved in the establishment of those marks. For the transcriptional regulators, well, as there's no major developmental phenotype, it's hard to say, because we cannot.
Like, for example, like if you look at the boundary in the meristem, right, the boundary specific gene regulators, like, would they, that's what I was thinking about on the right hand side, where in your picture you have the wild type and the crown mutant.
Yes.
I don't know enough about the transcription factor machinery involved in these apical meristem developmental processes, but if there were any that were preferentially expressed in the boundary, like, I don't know if STM is a transcription factor or some other type of protein.
Yes.
Yeah. Okay.
But like, of course, we could talk about the STM. We could talk about the CAC gene, but from what I saw, the morphology is not so changed. Like the boundaries, there's no phylotactic defects. There are no defects like splits of the meristem. So it would be extremely hard to quantify anything, and especially because the boundaries are still forming, and the nuclei are still being compressed in the boundary. So I would say it's would be really on the threshold of detection, if any. Because then,
yeah, thank you.
Yeah, so sort of related to that question, I guess I was thinking about this, what the physiological function is here, right? Because I mean, the data that you were just showing with the crown 4 mutant really uncouples nuclear shape from any developmental function, right? You can have totally messed up looking nuclei and your boundaries are fine and your like developmental phenotypes are fine, which makes me wonder if you've thought about all of these things you've seen going on in the boundary, where there's clearly a system set up to respond to the nuclear changes, that the histone stuff. Do you feel like what you're seeing in the boundary domain is sort of like a side effect, or like a sign that there's a system set up for responding to nuclear shape that's important somewhere else, and we just don't know where that is? Is that how you see it?
Well, not exactly. I would say that it comes back to the story with PAF1 complex, because that one is having something in the boundary, because the boundaries are not being set in the correct manner.
Sure.
And there are some splits, and it's about the transcriptional noise, and it's about the protection of the weak link, I think, rather than that, because when something is under stress, it has to be protected from making the mistakes. And maybe the cells in the boundary is about that. They have to be protected. There should be some mechanisms protecting the chromatin from making something which should not take place, to prevent those mistakes. And maybe the chromatin factors which are expressed in the boundaries, and or the compression of the chromatin and compression of the nucleus leads to that. And a biological function is to protect those boundaries, like protect the cells from proliferation, because they should not. The boundary has to stay without cell divisions.
Sure. I think, yeah, I agree with that. I guess I was thinking more about this idea that nuclear shape is a signal per se, that like nuclear shape, when you change the shape of the nucleus, that serves as a signal. That you kind of have to argue that's not the case, because you can really change nuclear shape and there doesn't appear to be a signal that is appreciable in these domains, right? That's all I'm saying. And I guess I was wondering if you had thought about, I don't know, other developmental regions of the plant that also show creases or or maybe convex instead of concave, like, I don't know where the lateral roots emerge, or the edges of the carpel where they bend in, and if maybe those would be places, or if instead we're just, this whole system is something more about like response to osmotic stress and not to these developmental domains aren't really relevant. I don't know.
Well, that's really interesting. But I was thinking actually about looking, for example, on a hypocotyl, because it's simpler as a system. It avoids all this developmental consequences because it's really early. It's just the seed is germinating and it's really fast in elongating. And so the cells which are there, they are subjected to the stretching. And as we saw in a crown mutant, they are disconnected. So they stay circular, and then this development still happens. So I would like to look inside those, and I would like to also test some the chromatin factors and their roles inside, and for example to challenge them mechanically and see what happens.
And doesn't the nuclear shape change along the length of the hypocotyl or something? Or maybe I'm thinking of the root. I'm thinking of the root.
Yes, they do. They have to be. They are elongated actually. They follow the general shape of the cells. There's a huge vacuole which pushes them on the side, but then the cells are elongated and the nucleus is following them. And there is also endoduplication which takes place inside. So there's the nucleus is increasing in size and increasing in size with the length of the cell, and so it's actually the whole system. But then it would be interesting to follow the time course. How is it following the elongation? And if we prevent the elongation, or if we use some mutants of the cell walls, which are not elongating the same way, what happens to the nucleus?
Yeah, that sounds really exciting. Okay, does anybody else have any more questions for Kateryna? I'll scroll through here really quickly. I don't see any. So I think we'll stop there, and just everybody, please thank Kateryna again for a wonderful talk. Very stimulating, lots of cool things for us to think about. So thank you.
Thank you so much.
Kateryna, again,